So as we know, the ticks and mosquitoes, they are blood feeders, and their blood feeder behavior actually is a long time history behavior. And as we can see from this news, they found that there is a 99 million year of ticks sucked the blood of fatteners. And also, another news that they found that the tick, a 46 million year old mosquito, contains blood. This is very impressive. And also, there is there's a wonderful name. Sorry, that is just an advertisement. So I'm not going to cover that. So anyway, so both ticks and mosquitoes, they have been started to evolve millions of years to have blood feeding adaptation. And my postdoctoral job is mainly focused on ticks, but in the future, I will also include the mosquito as well. So I believe all of you know ticks. So when you are hiking or walk through the grass, the tick might find you and search your skin, and then look at your suitable spot, and then cut your skin and then ingest the blood. And the right picture is the real picture I took 2 months ago. So one distinguished professor from Yale Immunology Department, he was hiking in New York, but he got 3 ticks biting in his back and then and the legs. So this is the picture I took. And this is very horrible. And this is the first time I've seen human being bitten by ticks, actually. And this makes me feel what I'm doing is really a big deal. And by the way, I have a question here. How many of you were bitten by ticks? Oh, what, really? God. Actually, this is not. Yeah, this is not expected actually. So how are you guys feeling? Did you guys test the infection status of ticks? You never tested.
Do, yes.
You do?
Yeah.
Not infected?
Oh, you mean specific ticks that did this? Anybody actually tested one of the ticks that did this?
No.
No.
That's good. Okay. Maybe in the future I can test the infection status of you. So the reason why we care about ticks, because ticks can transmit tick-borne disease, right? Such as the Lyme disease, as you know, and the babesiosis, and many fevers. And the right picture is another real picture I took last spring when I was hiking in New York, Bear Mountain. And we can see here is notice showing to warn people the dangers of Lyme disease. And we can also consider there is a small notice here that means plus bear live here. So no matter how big the bear is or how tiny our tick is, they are both dangerous animals, right? So regarding of tick feeding adaptations, and I listed some questions from this reference. And the first one is what was the nature of the ancestral tick, and when did the ticks evolve to blood feeding capabilities, and how did these capabilities evolve? And did the host specifically influence the adaptation of ticks to a blood feeding environment? And what are the implications of tick evolution for future research into tick biology and vaccine development? And also I would add the last one, that is the pathogens, that is a vector pathogen host evolution, co-evolution. And in this talk, I will cover these 3 questions through study of tick saliva vaccine studies. So when tick bite you or other animals, tick can secrete saliva, right? So tick saliva has evolved to have so many functions. The first one, as I mentioned, is blood feed adaptation. For example, the tick saliva has anticoagulation role. And also, tick saliva has pathogen transmission role. For example, there is a tick saliva protein called SUPR15. That protein can inhibit CD4 T cells activation, so that it can facilitate the pathogen transmission. And since tick has so many salivas, so many valuable salivas, and so many functions. So we have a question that's my first project, the question that can we develop the vaccine based on tick saliva? Actually, my postdoctoral advisor, Dr. Ira Figueroa, who is the person helped to develop the first FDA approved the Lyme disease vaccine. This vaccine is based on the Borrelia, the Lyme disease agent Borrelia, the OSPA, autonomous protein. And actually, unfortunately, this vaccine is not available anymore because of market demand. And now our strategy is to target a vector salivary protein. So by this strategy, supposedly we can broadly protect the vector disease, right? So we select 19 identified proteins as candidates. And then we use the guinea pig as the model to test that. So the reason why you use guinea pig because guinea pig has shown to have tick immunity. So tick immunity is if we put a second time tick on guinea pig, the guinea pig has tick rejection. It cannot feed anymore in the second time. So that we use guinea pig as a model. And we use the very advanced mRNA vaccine technologies. And we directly collaborated with Dr. Joe Wiseman, who is one of the pioneers of the mRNA vaccine technology. And in the last 2 years, my advisor always told us, if we want to synthesize tick mRNA vaccine, contact him before he got a Nobel Prize. So, and I believe he will get a Nobel Prize someday. And because of him, we are still survive after the COVID pandemic. So the basic idea is we put 19 tick salivary protein in the lipid nanoparticle, and then immunize the mice, the guinea pigs. So the guinea pig can develop the protein and generate an antibody by itself. So I was the one of the senior contributor in this project. So the idea is we inject the mRNA vaccine to guinea pig, and then we have 2 boosters every 4 weeks. And then after 2 weeks, 2 weeks after the final booster, we bleed the guinea pig to test the antibody titers of 19 proteins. So here is the ELISA data. We can see most of the 19 salivary proteins has much higher the antibody titers, especially SAP14, the PDI3, SG10, SG27. So after confirm this, we challenge the guinea pigs with the ticks. Here's the data. So we can obviously see there's a erythema started to showing as early as 18 hours in 19 ISP immunized animals. However, in control animals, there's no redness, right? And we also see that the ticks start to die or drop off from the immunized animals after 48 hours. But in control animals, the ticks feed very naturally. So this data suggests that target to the vector salivary protein is very promising for prevent tick bite in the future. And we also test if this vaccine can prevent the pathogen's transmission. So we select the Lyme disease agent Borrelia as the target. So as you know, the Lyme disease is a very big issue in the northeast part of the United States. And when tick bites you, the Borrelia can be injected to your skin and then disperse to distal organs such as heart and jaw joint to cause diseases. So we use Lyme disease as a target, and we challenge the guinea pig with Borrelia infected ticks. And we tested the infection status of the guinea pigs. As we can see, in control animals, the 46% of guinea pigs were tested positive for Lyme disease. However, 0% of guinea pigs were tested positive. So this data suggests that our vaccine also at least works to prevent Lyme disease agent. And this project has been published in Science Translational Medicine. We got the cover. And then I wrote the M2 for this project, R1IH R1 project, and it was funded with a top 7% score. And in the next steps, we are going to test this vaccine in other animals, such as rabbit, the deer, the monkey, as well as also humanized mouse. And I just show you some preliminary data on rabbit. So when we immunize the rabbit, we can see ticks feed on immunized rabbits has less reproduction. There is the X there, here's the ticks feed on immunized animals, that has less production. So this is a very important data. So if we validate this data in the field, it will significantly decrease the population in the field, right? So I'm very excited about this. So in summary, in my mind, the tick, what tick security is not saliva, it's gold. So this cellular protein has evolved to have many functions such as promote tick feeding or promote the disease transmission. But the major job we will do is to identify more salivary candidates. We just targeted 19 candidates, but there are more valuable tick salivary proteins. So the job we are currently working on is like, in my mind, is like gold miner. So if we push the play button and we go to the project 2 to play this game, and then, so in project 2, I identify a novel tick salivary protein, which is related to pathogen transmission. And the name of this protein is the complement C1q-like protein, as C1qr3, or we just call it tick C1q protein. And how did I identify this gold protein? I will keep it for secret just for 20 minutes. Don't be jealous. So interestingly, this protein has no any homologs in any insects, including Drosophila and mosquitoes. It has no homologs in insects, but it do have some homologs in ticks. And more interestingly, these homologs are only present in hard ticks, not soft ticks. So this data suggests that it could be an independent evolution of this protein, and only hard ticks evolved to have this protein for some specific purpose. And then I hypothesized that this protein was acquired from other animals or bacteria from by horizontal gene transfer, but this has not been validated yet. So if you guys are interested in this, I would be happy to collaborate to see what's going on there. The reason I said this is a salivary protein because from the gene expression, we can see this protein gene expression is much, much higher in salivary gland compared to mid-gut. And also this protein has a signal peptide. And then that's why I hypothesized that this is a salivary protein. And then we collect the tick saliva very quickly and then probe the saliva with C1q antiserum. And we do see the one band which is very close to the predicted molecular weight of this protein, that is 17 kD in reducing conditions. And also, I also probe the clean tick saliva and the Borrelia infected tick saliva, the same amount with the antiserum. And we can see the Borrelia infected tick saliva has much, much higher protein expression compared to clean saliva. This may suggest that the Borrelia can induce this protein expression. And then we further found that this tick C1q protein actually can directly interact with Borrelia spiricate. From the pour down, we can see that protein can bind to Borrelia pellet. And also from the flow cytometry, we can see compared to control, you can see this protein has some reactions with the Borrelia spirochete. And also from the ELISA data, we can see this protein interact with Borrelia in a dose manner, and the immunofluorescence assay also confirms the binding of TIC-C1q protein, which is green, with the Borrelia spirochete, which is blue. So since Borrelia induce this protein expression in tick saliva, and Borrelia can directly interact with this protein. So we have a question that whether TIC-C1q protein has a role influencing Borrelia transmission from the vectors to the vertebrate host. So then we first silence this protein gene in tick by injecting the double-stranded RNA, and then in infected ticks, and then we put the ticks on the clean mice, and then we test the Borrelia burden in mice skin at 7, 14, and 21 days. And here is the data, and we can see the mice fed by C1q protein gene silenced ticks has less Borrelia burden in mice skin at day 7. However, day 14 and day 21, there's no significant difference between these two groups. So this data suggests that the TIC-C1q protein may facilitate the Borrelia infection, early infection to mice. And then we further blocking this protein in tick. So we inject with antibody. So we inject the C1q recombinant protein into the ticks every 2 weeks. And then we put the infected ticks on the immunized mice. And here we found that all the immunized mice has significantly lower Borrelia burden compared to the control at day 7. This is consistent with silencing this gene. However, at day 14 and day 21, there is no difference. So then I trying to investigate the potential mechanism under this phenotype. So here I list some mechanisms or functions of C1q domain containing the proteins in membrane system. So as you know, the most famous function of C1q proteins are classical complement system. And the other functions like affecting the production of cytokines, and in most cases is anti-inflammation, and can modulate the phagocytosis and other many functions. So I started to investigate if this protein can manipulate host complement immune system. So if you are familiar with the mammal complement system, so the mammal C1q protein can bind to C1R and C1S to form a complex and then activate the complement cascade. So I first tested if our TIC-C1q protein can bind to C1R or C1S or not. So by ELISA, we can see there's no any interaction between our TIC-C1q protein with C1R or C1S. And also in the mammal C1q protein, the globular head domain, and our TIC-C1q also has a globular C1q domain. This domain can bind to IgG antibody FC site. Then I tested if our TIC-C1 protein can bind to FC region or not. However, by ELISA, I cannot see any interaction between C1q protein and the FC region. So this suggests that the TIC-C1 protein is probably not involved in complement pathway cascade. So then I trying to investigate if TIC-C1 protein has role in other immune system. So I select some representative works, the genes which are involved in pro-inflammation or anti-inflammation, and tested the gene expression after incubate with our TIC-C1 protein as well as BSA as the control protein, and then use LPS to stimulate and test the cytokine gene expression. And we found that the TIC-C1 protein, and I used the splenocyte, which is an immune organ in mice, and we found that TIC-C1 protein can significantly inhibit two cytokine gene expression. One is CCL3, which is related to macrophages, and another one is the interferon gamma. And then actually the interferon gamma can be induced by Borrelia in mammals. So then I use the Borrelia as the stimulate agent, and then incubate with the TIC protein, and then check the cytokine gene expression. So here we found that after stimulating with Borrelia, we found the TIC protein can still inhibit interferon gamma gene expression. However, for CCL3, we didn't see any difference anymore. So this is the data from in vitro. How about in vivo? So then I silenced the TIC1Q protein gene again, and then put it on mice and tested the cytokine gene expression in mice skin. And we can find that two cytokines, one is TNF alpha, and the other is interferon gamma again, is significantly upregulated in the absence of TIC1Q protein. So all this data suggests that TIC1Q protein has some relationship with the interferon gamma protection. Then to have a global idea of the TIC1Q protein effect on mammals, so I did the RNA-seq. And very interestingly, we found that most of the differentially expressed genes are the top genes I highlighted are all related to interferon gamma production, actually, such as interferon gamma induced the GPP proteins. There are many GPP proteins here, as well as the CXCL9, and also the majority of differential genes are highly rich in cell response to interferon gamma pathway. So this data further confirmed that the TIC1Q protein must have some relationship with the interferon gamma production. So then we have another question that so which immune cells were targeted by TIC1Q protein to regulate interferon gamma production? So as we know, that interferon gamma is preliminarily secreted by T cells, the macrophages, and the natural killer cells, right? So then we use the intracellular staining and the flow cytometry to find which immune cells category were targeted by TIC1Q protein. So then we first remove the beta cells. We only keep the liver cells, and then we separately getted the different immune cells categories, which are CD4 T cells, CD8 T cells, the macrophages, as well as the natural killer cells. And then we tested the interferon gamma production. And then we found that the TIC1Q protein can significantly inhibit interferon gamma production in CD4 T cells and CD8 T cells, but not in macrophages and the natural killer cells. And then we isolated the CD4 and the CD8 T cells to see if TIC1Q protein can really inhibit the interferon gamma in them. So we isolated them, and we found that after isolation, the majority of the isolated cells are the CD4 T cells and the CD8 T cells. The majority are there. And then we incubate with the TIC1Q protein and stimulate with Borrelia. And we further found that the TIC1Q protein can really inhibit interferon gamma production in isolated CD4 and CD8 T cells. So we have another question that how does TIC1Q protein regulate cytokine production? So what's the receptor on the immune cells? Actually, there, as I said, the TIC1Q protein has a global C1Q domain. Actually, in mammals, there's one receptor called the GC1Q receptor. This receptor has been shown that it has negative regulation of interferon gamma or IL-12 production. Also, many other pathogens like HCV virus can hijack this receptor and regulate some cytokines which include interferon gamma. So this receptor is a very candidate to work on, and very lucky we will quickly confirm that TYK1 protein can interact with this G1QR receptor by co-IP, ELASA, as well as immune co-immunolocalization. And then we trying to understand even they can interact. So this interaction can contribute to interferon gamma protection or not. So then we block. So we block this G1QR with this antibody to see if this blocking can affect TYK1 protein mediated interferon gamma protection or not. So first we tested this antibody on interferon gamma protection in immune cells. We found that at the concentration of 25.5 microgram per milliliter, this antibody binded to G1QR can significantly increase the interferon gamma protection. And then we incubate with the TYK1 protein and stimulate with Borrelia again. And we found that at the same concentration, 25.5 microgram per milliliter, after blocking the TYK1 protein has no effect on immune cells interferon gamma. However, has lower concentration, 2.55. The TYK1 protein can still inhibit interferon gamma in immune cells. Then we further silence this receptor by siRNA. And then after silence this receptor, we found that the TYK1 protein has no effect on interferon gamma in immune cells. So all this data suggests that the TYK1 protein can interact with the mammal G1QR on immune cells to modulate the immune responses, in particular the interferon gamma protection. So this is the mechanism. How about the application? So we synthesize the mRNA vaccine based on this novel TYK7 protein, and then I immunize the mice every 2 weeks, and then I bleed the mice, and also put the ticks the infected ticks to challenge the mice. And here's the ELASA data. We show that all the animals have high titers of the TYK1 protein, and also the Western blot further confirmed the specific activity. And then we found that the important data is here. We found that the immunized mice has less Borrelia burden in mice skin. And we did this experiment twice. So in summary, the TYK1 protein is a TYK salivary antigen and can directly interact with Borrelia spearkii. And the TYK1 protein contributes to the initial stages of Borrelia infection mice. And the TYK1 protein alternates the early host immune responses to infection of Borrelia by decreasing the interferon gamma through interaction with the global receptor present on the surface of T cells. And the mRNA vaccine decreased the Borrelia infection in mice. And I wrote the whole grant for this project, and we got discussed in the first round, but have not funded yet, and this grant is under review. So if next I will talk about my future work. The topic of my future work is evolutionary vector biology. And I know there are so many studies focused on this topic. As I mentioned, as I said, like Dr. Beatty and her postdoc just published the one paper this year, and they found they investigated the novel genome sequence and the evolutionary dynamics of the Anopheles mosquitoes. And there are also other papers like evolution and diversity of mosquitoes and the phylogeography dynamics of the ticks. But what I'm interested is the vector pathogen host co-evolution from a functional perspective. And here is the vector pathogen and host triangle. And sometimes the environment factors are also involved in this. So what I'm interested in is the vector pathogen host co-evolution. So I will first screen the vector pathogen host co-evolution or interaction components, and then investigate these components or proteins function and mechanisms from the evolutionary perspective. But the main job is how to quickly screen the vector pathogen host interactions components. So then, the best idea is that use the genomic and bioinformatic techniques, right? So how to do that? So I will explain in the next slides. So Dr. Aaron Lin, who is the assistant.
Are we having lunch?
In immunology department.
It's past 12. It's 12:10.
Construct the humans.
Not always. It's just because I was staring at screens too much.
So by this way, we have identified.
Okay, I'll ask Jen too.
Proteins can interact with pathogens, any pathogens, and then by a protein assay, and they identify the bound protein by unique markers. So then his idea is just express the members protein or secret protein on the yeast library, on the yeast actually. So by this way, he conducted the yeast library. Actually, we have a trial showing that we use the Lyme disease circuit to probe this library. And I also involved in this project. Right? So the first study is that we found that based on this library, that human protein, that the PG recognition protein 1 has highest score to binding with the Lyme disease agent circuit. And this binding can influence Lyme paralysis. And another study is that we use the relapsing fever agent, the Borrelia culture, right, to probe this library. And we found that human CD55 can highly binding to the relapsing fever agent. So use the same. So this is the host pathogen interactions. How about the vector pathogen interactions? So use the same strategy. We can screen the vector pathogen interactions, right? So here I just give you an example of the Aedes aegypti library. So based on, so after a few steps of bioinformatics tools, so we can identify as much as the secret mosquito protein or mosquito members protein, and then we synthesize the plasmid that contains those sequence, and then we express on the yeast members, and then we probe the pathogens we like. So by this way, we can high throughput screen almost all the interactions network between vector and pathogens. And we are currently working on the DRTIC library. We also work on Aedes library and Anaphylaxis library. And I have talked with my postdoc advisor, and he said I can take these libraries with me if I start my own lab. So this is just kind of collaboration work. But for independent work, as an independent principal investigator, I will talk about the pathogen vector interaction and also pathogen host interaction. But how about the vector host interactions, right? So in the future, I will focus on the evolutionary cross-species interactions. I mean, I will focus, I'm more interested in host factors. And as you know, the most common host factors interact with the vector is blood. So both ticks and mosquitoes has a long history of blood feeding, and they have blood feeding adaptations. So the blood can stay with the vector gut, especially gut, I mean, very frequently, right? So there must be, from a long history, there must be some interactions cross-species or cross-kingdom interactions between the blood and the vectors. And I just take the Dr. Chloe Hawkins and Grayson's marriage as an example. So they were both studied in Texas and AM before. So they're both on the 5th floor of our entomology building. so they can communicate and talk and chat every year, every week, every second, every minute. So after a long time, I mean, just a couple of months. After a long time, there must be some reactions between them. So at the end, they got married. So from my perspective, their marriage is evolutionary consequences. Just kidding. And yeah, so for this topic, I will actually in ticks, there's one example showing that the host factors, like what I said, is the cytokine interferon gamma. This study shows that the interferon gamma from incoming blood can interact with the unknown receptors to regulate the tick stat or TTBS signal pathway to regulate the antimicro peptides DA2, which is limited to earlier survival. Right? So actually this year, very early this year, January 13, they found the unknown receptors. That is number 1. So they found that this signal pathway can regulate the vector immunity and development at the same time. Actually, I'm started to interested in this topic from my beginning of my postdoctoral training. So I have finding that the host factor, the host blood component, adiponectin, can cross-speciously interact with tick adiponectin receptor. So this cross-kingdom interact and regulate tick glucose metabolism. So the reason why I focus on adiponectin, because adiponectin has high concentration in blood, is like 5 to 30 microgram per milliliter. And the main function of it is fatty acid oxidation and glucose and lipid metabolism. So that's why I start to work on this. And I also found that, remember I said I keep that protein as a secret for 20 minutes, is here. So actually this cellular protein C1q is the homolog of adiponectin. Yeah, adiponectin is also a C1q domain containing protein. So we found that the Borrelia can co-opt this C1q protein to regulate the tick phospholipid metabolism. And phospholipid metabolism is very critical for Borrelia mammals synthesis. And during review, I don't know who is the reviewer, suggests us to cite this reference, which Dr. Kalada is the main contributor. I'm not sure the reviewer is you or not, to cite this publication. And this publication actually is very support our finding, because Dr. Kalada found that there is a lipid exchange between Borrelia and the host cells. So, and it's greatly supported my finding. And we also found that the Borrelia can co-opt tick phospholipid metabolism to facilitate its mammal synthesis. Okay, so I just give you this one example. And recently I have a new novel data showing that the ticks, the NIFS ticks, feed on what type of adiponectin knockout mice. We have knockout mice. And then the ticks feed on knockout mice has significant lower molting rate. And also based on RNA-seq, the mammal adiponectin can regulate multiple metabolism pathway, like especially the nutrients molecular transport, such as the lipid chemical process, the sodium ion transmembrane transport. So, this pathways transport metabolism molecular transport pathways are highly reached. So this means the mammal admitting may affect tick metabolism. And also I have another data showing that admitting can affect Borrelia transmission by ticks. Actually, here we found the admitting knockout mice has significant higher Borrelia burden in the early infection. So in the future, I'm currently working on why and how admitting regulate vector immunology and development from a co-evolutionary perspective. So in summary, my future work topic is I will continue work on evolutionary cross-species interactions. So I will mainly answer three questions. The first one is there any more components in mammals, or especially the blood components, can cross-species talk with blood feeders? So I will use the blood components such as the cytokine, the hormone protein to probe the vector yeast library, which I can tag with this library. So can highly throughput screen as much as interaction components networks between the host factors as well as the vector. And then we investigate the mechanism from the evolutionary perspective and how can cross kingdom interaction happen. It is vector driving or it is pathogens co-opt this. And also the outcomes is the cross-species interactions may affect vector development as I just shown, and it also can affect vector combinance or pathogen infectivity, which I also show in the preliminary data. So this is a very promising, very interesting. I'm very excited for this topic. And I will work on the vectors like Scapularis, and especially the Asia longhorn ticks. And actually, this ticks is the first found in New Jersey. And if I remember correctly, the James O.C., if I pronounce correct.
Raining right there.
Oh yeah, you are the main contributor for finding this. From sheep, yeah. So I will work on mosquitoes as well to to screen as much as components and investigate the mechanisms from an evolutionary perspective. So future work as a summary. The first I will use genomic and bioinformatical techniques to high throughput screen vector pathogen host interactions. And I will investigate the evolutionary forces in addition to mutation, gene flow, gene draft, or natural selection. And I will focus more on vector drawing. Is vector drawing or pathogen drawing or host drawing for its unknown purpose? So there is too much work we need to do. And also, I will get the mechanisms and more candidates for vaccine development, which is my expertise. And the second one is evolutionary cross-species interaction studies. And by this study, I can get the mechanisms and novel signaling cascade for disrupting the vector development and pathogens like SCUR. And I will conduct my studies at multiple scales, like the molecular, organizer, population, community, and regional scales. So in the end, I would like to thank all the members in my current lab. And our lab is a very big lab. It's like close to 30 people, and we have 20-something postdoctors. So our lab is INIH funded, the Cohen Foundation funded, and the HHMI funded lab. And my advisor is HHMI investigator. So in the end, I would like to mention that the love between Chloe and Grayson is a non-cross species love. So, and with that, I would thank you all of you being here, and I'm glad to take any questions you may have. Thank you. So, questions.
Well, I have a question.
Sure.
So I have a bunch of questions, but sort of
we can talk about.
yes, but so what are the aspects of what you're doing is basically I'm not a vertebrate immunologist, so some of the information you've covered is kind of goes over my head. But one of the things that seems striking to me is that you seem to be slowing down the transmission of borrelia burgdorferi,
Yeah.
not necessarily stopping.
Yeah, it's partially stop. It's not completely stop, right?
So what's the consequence of that? I mean, if you instead of injecting X number, you inject X minus number, what's the effect from the disease then?
Yeah, so I will try to answer this question as far as I know. So the job I did is just the candidates I did, of course, is just partially protect mice from or other animals from infection. So the main job of what I emphasize is that we need to screen more cellular proteins, any vectors. So there are like 1,000 something cellular proteins, and in the first project we only targeted 19.
Right.
So there's so many valuable cellular proteins to identify. That's too much work to work. And then we use the cut edge, the Eastern library to screen as much as we can. So if we are lucky, supposedly, if we can identify almost the valuable cellular proteins in any vector, supposedly we can completely prevent.
So it's like a cocktail.
Yeah. Actually, 19 proteins is the cocktail vaccine. Yeah. So that's too much work. We need to work on this. That's our job, right? A vector biologist. Any? Let me show. Oh, should I stop the record?
You can stop sharing unless people have.
Is there any question? Oh no. Okay. You are record. Okay.
Okay, Anne.
Oh, hi.
Far away from I'm not from Quebec, but I know it's me.
Yeah.
So about how long away from
Oh, we just started.
Could you repeat the question?
Oh yeah. So sorry for the Zoom people. So the main question is how far away from the natural vaccine can be FDA approved to the market and sell in the market. So we are just started actually, and we're trying to use different ways. And actually, that's my postdoc advisor's dream to develop a vector, is in particular the ticks vaccine. So that's why we're trying to use the cut edge the vaccine technologies like the mRNA vaccine we did. So this is the most advanced actually the vaccine technology in these days. So actually we are very fortunate that we use the guinea pig as the model. We found that this vaccine is very efficient in the guinea pig. We didn't test the human yet, and also dogs, cat yet. So but that's our future plan and we have the preliminary data on rabbit. So if that data can be validated in the field, we can significantly decrease the tick populations, right? It's less, if the ticks in the field feed on immunized rabbits or other animals, if this vaccine works, it's very few eggs. So the ticks population significantly decreased. And we are just a starter from this, but we think we are in the right track to develop the natural vaccine, which can be approved by FDA. Yeah, Doctor.
So guinea pigs are kind of resistant to tick bites, right?
Yes.
So why didn't you choose a rodent model that reflects better the life cycle of the tick rather than because if you want to prevent tick populations and not preventing those ticks from acquiring diseases, right? The main reservoirs for those are going to be mouse, field mouse, right?
Yeah. So yes.
Why didn't you try field mouse, which is more relevant? So at least model use rather than.
Okay. So the question is why we didn't use the fed wild animal mice or other animals. Why we focus on guinea pig, right? So the guinea pig has the tick immunity. The mice don't have, in particular for its capillary ticks, that they have no tick immunity. So in the lab, we can feed the mice with ticks multiple times to do experiment. So that's the main reason we use the guinea pig as a model from the beginning. But I also mentioned this talk, we are using the humanized mice. Human has tick immunity. We use the human, so the humanized mice is most similar to the human immune system. So we are trying to use the humanized mice. So Yale has a good labs for develop the humanized mice. So we are working on that, but not finished yet. But so if the humanized mice have tick rejection, wow, we have too much work need to in the future, because guinea pig is not a model animal, right? Mice is a model animal, so we can develop a mice have tick immunity, because we have so many mechanisms to discover. So too much work to do that. That's our another goal. So, yeah, did I answer the question?
I have another question, just
Sure.
because you keep making parallels between what you're doing on pigs and like mosquitoes. For example.
Yes.
In the first example you gave of the first vaccine, the vaccine that is closest to release, you basically were able to have a tick not be able to feed after about
48 hours.
14 hours. Mosquito takes 2.5 minutes.
Yeah.
To feed. So how will this be later?
Oh, okay, that's a very, very good question. So actually, our lab is also develop the mosquito salivary protein vaccine as well, MR vaccine as well. So the main purpose, the main goal we did is not to prevent a mosquito bite you. Mosquito can still bite you. The main purpose is to disturb the virus transmission. Yeah, that's what we did.
Just because that's obvious.
Yeah.
What you need to do.
Yeah.
So
So
focusing on the pathogen.
yeah, only focus on the pathogens. And actually, our tick vaccine, you see the erythema, so it looks like the mosquito bite erythema. So if imagine if people have vaccinated by 19SP, our vaccine, the erythema will generate very quickly, maybe 18 hours. And people will see this erythema and remove ticks. They thought, oh, I got a tick bite.
Because they noticed.
Yeah, notice it.
Yeah.
It's another advantage of our vaccine. Yeah.
Yeah, and by the way, I should just also say, just to everybody, that most people here that got bit by a tick detected
Not
it early
exactly.
enough that they knew there would be no transmission. So that's probably why they didn't test.
Ah,
But I just want to have a disclaimer and everybody.
yes. So
Because
our
we're good at detecting them. So
our
although we
normal
were
vaccine.
bit,
Yes.
we knew that we caught it early. So just
Mhm.
now, clear that. You have a question?
Yeah, sure.
The 19 proteins that you do find, I think,
Mhm.
I'm assuming that they were all focused on Ixodes scapularis, but are there homologs in other ticks?
Yes, that's a very good question. So the answer is yes. And we have an experiment actually. I have a colleague, and he already left. So he tested the cross species, not interaction, sorry, cross species protection. So he using the scapularis feed on guinea pig first. And then the second round, he put the another ticks, I forgot which ticks, sorry about that. And actually that ticks has tick immunity. So if you immunize with the scapularis saliva, it can protect you from other ticks. That's our data in the field, in the laboratory. It can. In the laboratory, but I'm not sure in the field it can do this or not.
Right, it's 12:30.
Oh, really?
So, yeah, no,
It's so nice talking with you.
Any other questions? And I know we're all going to be meeting, students,
Right.
lunch or food. Well, thank you very much.
Thank you. Thank you for coming. I appreciate it. Then I stop. So you can stop a record first, and then
Yeah, you can.
I can stop
you
share.
can just end.
Okay. Thank you, everyone.